Welcome to the HPL seminar this week. It is my pleasure to introduce today's speaker, Tim van der Zee. Tim started his undergrad in the program of Human Movement Science at Vrije University, Amsterdam in Netherlands. His undergrad project was specialized in biomechanics, which was specifically about how to predict metabolic energy cost using Hill and Huxley type muscle models. Then he got his bachelor's degree in 2016 and started his master's course in the next year at the same university under the supervision of Dr. Nook van Swisht. His research project was to investigate the metabolic cost of constant muscle force production and achieved his master's degree in 2018. Then he came to the University of Calgary in 2019 and started his PhD in biomedical engineering, working with Dr. Art Kuo. Tim has been trying to link muscle mechanics to energetics by now specifically working on his thesis project addressing the metabolic cost of fluctuating muscle force production. He has been also interested in understanding the role of muscle in human walking. So he is planning to further work on creating a mechanistic muscle model that can predict the metabolic cost of locomotion in the future. In addition to his academic background, Tim gave me an interesting fun fact about himself that he loves biking. And he also mentioned that he even biked from Paris to Corsica in France in 6 days. But I wasn't sure what this exactly means. So I searched the map and realized that it was over 1,200 kilometers in 6 days. Then I got impressed. Today, he was going to share part of his thesis work entitled as High Energetic Cost of Rapid Force Development. All yours, Tim.
Thank you, Sangwa. I'll time it. All right, so I'd like to start with two muscle energetics mysteries. The first is that humans walk in a stereotypical manner. They walk at a certain speed with a certain step frequency. And the second is that rattlesnakes rattle in a stereotypical manner, and they choose a certain rattle frequency. And here in this talk, I will speculate that both may be related to the same hypothesized mechanism, which is the force rate cost. So a little bit more about human walking. Humans adjust their step frequency to their speed. But why do they do that? Why don't they just choose a constant step frequency? Having a high constant step frequency may be beneficial for reducing step length and thereby reducing the knee joint or the joint moments. Rattlesnakes, they increase their rattle frequency with body temperature. But again, why do they do that? Using a high rattle frequency at all temperatures may be beneficial for them because it increases the noise of the rattle. So we have these two phenomena that we don't really understand. And here in this talk, I will argue that both may be related to the force rate cost, that rattlesnakes rattle at a low frequency at low body temperature to avoid the force rate cost, and that humans step at a low frequency at low speeds to reduce the force rate cost. Before I will explain why force rate has a cost, I will give a definition of force rate itself. Force rate is the time derivative of force, which you can visualize as the change in force divided by the change in time. And this becomes a derivative if that change approaches zero. In this talk, I will start with giving a mechanism for the force rate cost. First, I'll do that using a qualitative cartoon and then using a quantitative model. And then I will go into a human experiment designed to determine the force rate cost. This is the cartoon. It's a cartoon of a muscle fiber. In the muscle fiber is the sarcoplasmic reticulum that contains calcium. The sarcoplasmic reticulum has a calcium pump, and that pump can pump calcium to the sarcoplasmic reticulum so that in rest the amount of calcium in the myoplasm is small. And this pumping requires the energy stored in ATP, as 1 ATP is consumed per 2 calcium transported. The sarcoplasmic reticulum is connected to the sarcolemma with some channels. And then, of course, we also have the myofilaments. And the link with the calcium is that they need calcium in order to interact and produce force and do work. If we want to produce force, we have to start with a muscle action potential. The action potential can travel down the sarcolemma when it hits the channels. This triggers the release of calcium into the myoplasm. Calcium can then reach the myofilaments, which can then interact. The cross bridges, so to say, they also consume energy, 1 ATP per stroke, and this is done by myosin ATPase. And note, well, this cost is then typically associated with work, as some work is done during the stroke. And note that this is a different cost than the calcium pumping cost, and it's the calcium pumping cost that we associate with force rate. All right, so we go from the action potential to myoplasmic calcium with some dynamics, calcium dynamics, and then from myoplasmic calcium to muscle force with some cross-bridge dynamics. And now I'd like to ask everyone to think about the question, which of these two dynamics is faster? And just make up your answer for yourself. I'll give you 5 seconds. The right answer is calcium dynamics is faster. We know this from measurements of calcium and muscle force. The reason why this is important is that from these experiments, typically the contraction dynamics are pretty slow, at least for slow twitch fibers. And that means that if we have a briefer force, we need a briefer myoplasmic calcium, but this also has to be of a higher amplitude. And this will mean that more energy would be required for the pumping, and I will explain this later. And the reason why I asked the question, so the implication is that we think that more ATP is needed for this, and this will increase with briefer forces. And now the reason I asked this question is that we found out that heel-type muscle models seem to have this wrong. In heel-type muscle models, we go from a stimulation via activation dynamics to activation, also called active state, and then from activation to muscle force using force length velocity. Force length velocity is quite fast. And the reason to understand this is to think about an isometric contraction at optimal length. In this case, the velocity is 0, the length is optimal, and therefore the force length velocity in the heel-type model does not have any dynamics anymore. And that means that there's just scaling between activation and muscle force, and this is instantaneous and therefore very, very fast. There is some dynamics with the activation dynamics modeled in these heel-type models, and that makes this the other way around. So slow first and then fast, instead of fast first and then slow. The implication is that for a briefer force, we now need briefer activation, but no increase in the amplitude, and therefore no increase in the cost. And also note that calcium is not explicitly present in the heel-type model in this form. And therefore we discard the heel model and start working towards a new model. So why does briefer force require more calcium? In order to explain this, I will focus on the rate limiting step between myoplasmic calcium and muscle force. I simplify this as a first order dynamics system. For those of you who are not familiar with first order dynamics, think of this as a low pass filter. If you now in this system use a step input, which is then a step in calcium, and look at the force will look like this. Now, if we use a step of the same amplitude but shorter duration, the force will look like this. And now note that the force is briefer, but it's also smaller in amplitude. And so there's a force difference. And we may not want that force difference. We may want to contract at a certain force amplitude. We can increase the force amplitude, but then in order to achieve that, we have to increase the calcium amplitude. So we need a higher calcium amplitude for the same force amplitude. And physiologically, how this can happen is by increasing recruitment, so increasing the amount of fibers involved. And it has also been speculated that using a doublet, that there's two action potentials shortly after each other, may increase the calcium concentration. And so, yeah, we have this calcium difference between between a briefer force. And in our cartoon, this looks like this, where we have, with the low calcium on the left, few calcium ions outside of the sarcoplasmic reticulum, and a lot of calcium ions in the myoplasm on the right side. And if you now think about cyclic force production, after every contraction, we have to relax and pump the calcium back. And that means that when the concentration of calcium is higher, we need to pump back more calcium. And this would require more metabolic energy used by the pump. And so we believe that the ATP cost per contraction should depend on the myoplasmic calcium concentration. And now we can quantify the difference between these two forces with the force rate, as the change in force is the same, but the change in time is different. So high force rate forces require more energy, and therefore we propose that the ATP cost per contraction should relate to the force rate. And this is the conclusion for now, is that the dynamics between calcium and muscle force imply an increased calcium amplitude with force rate. And this is associated with additional energy cost, the force rate cost. And we note that this is not predicted by Hill-type muscle models that have the wrong dynamics. Okay, this was all a little bit oversimplified maybe. So now I'll go into a quantitative model that hopefully is more accurate. So this was our simple model where we had a step input and a response. We can compare this to empirical data on the right from fluorescent calcium measurements. And then the force actually is not that bad, but the calcium is. If you compare in the upper right plot, the blue against the black, you see that, well, they both increase rapidly, but the black, the empirical measurement, also decays rapidly. And so there's something missing. And the thing that's missing mostly is that there's an extra step in between. And that's the step of calcium binding to troponin. And then this is our complete model where we start with the action potential. And this model is very similar to the Keldyck's model by Baylor and colleagues. And how Keldyck's works is that calcium is modeled either as and is either as occurring freely or is bound to a certain site. And the amount of bound calcium versus free calcium depends on the concentrations and on the forward and backward rate constants, K+1, K-1. In the Keldyck's model, there are also buffers. So calcium can also bind to these buffers instead of troponin. And there's a calcium pump. And these are some typical results from Baylor and colleagues, where we see that the calcium on the upper right is very brief, but actually the calcium troponin on the lower right increases very rapidly, but decays pretty slowly. And I replicated this model in MATLAB, and my results are very similar to their results. Note that the time scale is different, but if you have the same time scale, they are almost identical, showing a brief calcium, free calcium, and a calcium troponin that's longer lasting. Okay, well, this is pretty complex model, and for some purposes, we may want to have a simpler model that is as simple as the hill type model, but as the right dynamics. And so therefore, I fitted a simple model on this more complex model. For the free calcium, I used exponential approximation, and on the upper left, we see that the approximated calcium in red is pretty close to the actual calcium of the complex model. And then I used first order dynamics between free calcium and calcium troponin, and I fitted the time constants as to replicate the calcium troponin of the complex model. And the lower left, you see that the calcium troponin of the simple and complex models are almost identical. And this was accomplished using a low time constant for activation and a high time constant for deactivation. And I use the same first order dynamics with muscle force. Okay, well, now I've used a simple model to go back. So you specify a certain force that you need for a certain movement, and think about which calcium you need for that. So the force I quantified using a Gaussian with a mean, standard deviation, and an amplitude. And here I also did that for the calcium. And now you can do this for this force, but you can also do that for a different force that is briefer. And the briefness of the force here is specified by that standard deviation. And our hypothesis would say that we would expect a briefer calcium with a higher amplitude. And that is that calcium difference that is then thought to be underlying the force rate cost. And then we can quantify the difference between the forces using the force rate, as the change in force is the same, but the change in time is different. And these are some results from this simulation, where we start with a force with a high standard deviation. Note that the calcium is very similar to the force, but is a little bit leading it. On the right, we see the amplitude of both signals against the force rate of the peak force rate of the force signal, and both amplitudes are very similar. If you now decrease the standard deviation of the force becomes briefer. And what starts to happen is slowly the calcium amplitude starts to increase. And this is happening despite the force amplitude staying constant. And if we look at the extremes, then clearly there is a calcium difference despite the force amplitude being constant. We may simplify this just using a linear, and so see that the calcium amplitude increases with the force rate. Remember that this is done using a time constant of 50 milliseconds for the contraction dynamics. And this is dependent on that time constant, on the dynamics. If we choose a smaller time constant, the size of this effect is smaller. Okay, if we go back to our cartoon, the calcium difference comes at a cost. And that's because in cyclic force production, all the calcium needs to be pumped back. And the more calcium is released, the more calcium needs to be pumped back. And that's, again, substantiating our hypothesis that the cost per contraction increases with the force rate. So now our summary is this. We now know that the dynamics between calcium troponin and muscle force are responsible for the increase in calcium amplitude with force rate. This is associated with additional energy cost, the force rate cost. And yeah, reiterate that this is not predicted by the hill type muscle. And this is the end of the mechanism part. And this is all theoretical. And of course, at some point, we have to come up with some data. And this is what I'm going to talk about now. I'll first quickly discuss what has been done before and what are the issues with previous studies. Okay, this is some evidence for the force rate cost. Firstly, in isometric conditions, intermittent contractions or cyclic contractions have been found to have a higher metabolic cost than continuous contractions. And also, if the intermittent contractions have shorter duration, this comes at an even higher cost. And this is thought not to be explained by fiber type because these experiments have been done with electrostimulation that doesn't allow for different fiber recruitment. And it has also been done in muscles that have uniform fiber, so that consist of 90% of the same fiber type. But the issues with these studies is that they haven't parametrically varied force rate and investigated the effect on metabolic cost. And therefore, yeah, we cannot use them for testing our hypothesis. And then some studies from our lab have done that, have varied the force rate. And an example is the leg swinging study, but there are also some issues with this. In the leg swinging studies, participants were swinging their leg about their hip joint and matching a target of swing amplitude and frequency. And it was found that the metabolic rate increased with frequency and force rate also increased with frequency. And therefore on the left plot, we see that metabolic rate increased with force rate. On the right plot, you see that the work rate was constant. And so work was thought to not be responsible for this increase. However, this is the joint work rate. That is different from the muscle work rate. And that's because the muscle can do work against the tendon or against the series elastic element. And this does not have to affect the joint work. So we can have zero joint work, but still have non-zero muscle work. And the muscle work may be estimated from ultrasound, but this was not done in this study. And therefore, a big part of my study is estimating this muscle work. And then remember from our cartoon that muscle work has a different mechanism than the force rate cost. And this is why it can be a confounder. And that's why we'll move on to my ultrasound method that is aimed at estimating muscle work. This is a video of the vastus lateralis contracting during isometric torque production. And this is a case where this shortening is only done against series elastic elements because the joint angle is constant. In this image, we can see fascicles running sort of parallel at an angle, the pennation angle, between two aponeuroses. The fascicle length has been estimated before using a semi-automated algorithm called UltraTrack, but I didn't use this algorithm because I found that there were issues with it. Specifically for the vastus lateralis, you need to extrapolate a lot outside of the image frame, and this was not so easy with this algorithm. And there were some other issues, namely it's history dependent, and therefore you don't get a unique fascicle length for a given image. It cannot be automated, fully automated, because it requires manual fiber selection and drift correction. And it doesn't allow for aponeurosis detection. And yeah, because of these issues, I decided to make my own algorithm. And this is very different, my algorithm. My algorithm has two steps, well, two parts of it. The first part is meant to identify the aponeuroses. And the second part is meant to identify the muscle fascicle. And we'll first talk about the aponeuroses. So this is the raw image. The first step in aponeurosis detection is filtering. Here I used a Hessian filter aimed to detect the thicker lines in the image. And there are some boundary effects, so I cut the edges. Then I do thresholding. so that there's only black and white pixels. And then I split the image in two, where the upper half is associated with the superficial aponeurosis and the deep half with the deep aponeurosis. In each half, I then define the aponeurosis object as the longest object. And the aponeurosis, superficial aponeurosis, is then the deep edge on this longest object. Similar for the deep aponeurosis, and the deep aponeurosis is the superficial edge of the deep object. And these are the identified aponeuroses in the original image. And from these objects, we may determine the muscle thickness. And this is an important variable for the fascicle length estimation. Okay, then we go to the second part, which is aimed at the muscle fascicles. The first step is again filtering, using the same filter but with different settings. Now we filter the thinner lines in the image. And then the Hough transform is used to determine the angle, the most frequently occurring angle within a specified region. This is a little bit complex, but I will just go through it and hopefully you'll understand some of it. So what happens is you define the origin, for example, in the upper left. And then for each pixel in the image, you do an analysis of which lines fit through it. So for example, this white pixel, we see, we fit a line through it. And for example, this line, and we can quantify that line as the perpendicular distance to the origin and with the angle of the line to the horizontal. And the distance depends on the angle, as the distance is 0 for this second angle. And we can try all the different angles through the line, through the pixel, and then we can get a relationship between the distance to the origin, rho, and the line angle, specific for this pixel. We can do this for a different pixel. We can fit the same line, but also all the other lines, all the other angles, and they have different distances. The common line that goes through both points is the intersection in that distance angle plot. And now if we do this for all the pixels in the image, we get a lot of these sinusoidal looking lines. And you can make a heat map of that. And the heat is the amount of intersections between the lines, with white corresponding to higher numbers and red to lower numbers. And so the most white lines there, or points there in that image, is the most frequently occurring angle that has the most intersections. And this is the fascicle angle. This is a little bit complex, so don't worry too much about it, but it gives us this angle of the fascicle. And this is the angle with the horizontal. And then previously we had the thickness from the aponeurosis part. And with the fascicle angle, we can determine the pennation angle, which is the angle of the fascicle with the aponeurosis. And then the fascicle length follows from geometry, where it's the thickness divided by the sine of the pennation angle. And this is the actual data for isometric knee torque production. So that means at a constant knee angle with increasing knee torque. So knee torque is on the horizontal axis. And on the left, we see fascicle length. The fascicle length decreases, and this makes sense because increasing knee torque would require increasing muscle force, and therefore the series elastic element would be stretched. And because the total muscle tendon complex length is constant, this would mean a decreasing fascicle length. And on the right, with that pennation angle, we see that this is partially accomplished by increasing the pennation angle. Okay, now think of this drawing here, where the muscle fascicle is in series with a series elastic element, and the total length is constant. But because of the pennation angle, with the total length, we mean actually the series elastic element length plus the projection of the fascicle length on the horizontal. And the projection is equal to the fascicle length times the cosine of the pronation angle alpha. And this is constant. And therefore, the series elastic element is equal, or the change in series elastic element length is equal and opposite to that projection, the change in that projection. And therefore, using fascicle length and pronation angle, we can estimate the change in series elastic element length. Knee torque can be used to estimate the series elastic element force as by dividing it by moment arm. And moment arm is used using the tendon excursion method. And this gives us then the series elastic element force length relationship, or stress strain curve. And this stress strain curve can be, or it's actually a force length curve, but it looks similar. It can be used to determine the work. And this is the muscle work. And that's because the joint work is 0. This is isometric contraction. And then the muscle is only doing work on the series elastic element. And these work terms are equal and opposite. And the series elastic element work is equal to the series elastic element force integrated over the length change. And that's the area under this curve. And that is how we can estimate the muscle work. Okay, we use this muscle work in order to estimate the work cost and to see if the total cost is explained by work or is also or force rate is also required to explain the total cost. And this is determined in a cyclic force production experiment. This is the task: cyclic isometric torque production. Cyclic in that torque varies between 0 and a certain value cyclically. And this is done to manipulate the force rate. And isometric in the sense that the joint angle is constant. And this is done to facilitate the work estimation, as I explained. This is the setup. And note that the knee angle is actually closer to extension than shown in the video. The video was just a pilot, and this is the actual experiment. And the cyclic torque production was accomplished through showing the participant the torque target on the monitor in front of the chair and asking the participant to match this torque target. The participant was exerting torque with 2 legs, and this torque was added. And this is the torque target, which was a sinusoid at variable frequency. The amplitude of the sinusoid was equal to the offset, and that means that the range is between 0 and 2 times the amplitude. And so these are always positive, and this corresponds with extension. The frequency of the sinusoid determines the time between, well, 2 consecutive peaks. Okay, so the sinusoids at constant amplitude and variable frequency. The constant amplitude resulted in constant average torque across waveform frequencies and constant work per contraction, as the work per contraction then determines on the maximal force in that contraction, and that depends on the maximal torque. The force, the torque rate increased linearly with frequency due to the increasing frequency, and it was assumed that this resulted in a linear increase in the force rate, assuming a constant moment arm. The hypothesis is that the force rate cost is proportional to force rate, and because force rate was proportional to waveform frequency, we can test the prediction that force rate cost is proportional to waveform frequency. The force rate cost is a cost per contraction. As I said, after every contraction, you need to relax and pump the calcium back to the sarcoplasmic reticulum. The frequency sets the amount of contractions per second. And therefore, the force rate cost per unit time in watts, for example, is the force rate cost per contraction times the frequency. And this therefore is proportional to frequency squared. Similarly, the work cost per contraction is constant due to the constant amplitude, but the work cost per unit time would increase with frequency. Okay, and these two costs have two different underlying mechanisms. Myosin ATPase for the work and calcium pumping for the force rate. And therefore, we assumed we can add these costs to form a prediction on the total cost. And then there's actually a third cost, and that's the cost proportional to force, which has been related by previous work to the tension time integral, for example. And this cost is constant because the average torque is constant. And this acts as an offset in the total cost. So we have these three costs that give us a prediction on the total cost. We can also visualize it like this, where the total cost is made of these three parts. We can measure the total cost, or we measured the total cost using respirometry. And as I explained, we estimated the work, and thereby we can estimate the work cost. And the other two costs we can determine from regression. This is the work cost. We already have seen this data because the ultrasound experiment was done with the same participants at the same knee angle as this experiment. And so this is the force length curve for these participants. And what we now do is we determine the peak force in the cyclic force experiment, which is shown here, and then integrate until that peak force. And then this gives us a work estimate of approximately 1.2 joules per lag. Because there were two lags involved, both exerting the same force, this gives us an estimate of 2.4 joules. But this is the work, and we need to determine the work cost. And therefore, we need to assume a certain efficiency. We assume 30% efficiency, which gives us the coefficient to multiply the work. And as an example, at 3 Hertz, this would result in 24 watts of work cost. Okay, now I'll show the total cost measured from respirometry. And that was actually substantially more than this 24 watts. And this is already therefore an indication that the work cost cannot explain this total metabolic cost of cyclic force production. And now we move the work cost to the left side of the equation and determine the force rate cost and the force cost from the non-work cost using regression. And this gives us these curves. And the total curve fitted the data well with an R squared of 0.66 and was the coefficients were significant. Okay, you may think, why not just fit a straight line through this? That might be a lot easier. Well, this would not be appropriate, and that's because at 0 Hertz, that means constant force production, there is a metabolic cost, and this is the force cost. And we actually measured this cost in the same participants. And this is constant torque production at the same average torque as during the cyclic force production. And this was slightly higher, in fact, than the force cost estimated from regression. We also tried to predict this data using a metabolic hill type model. And the hill type model only predicted 7% of the increase. And this is in line with what I mentioned earlier, namely that the hill type model has the dynamics wrong and therefore cannot predict this force rate cost. Okay, so at 2.5 hertz, the force rate cost and the work cost were actually very similar. The metabolic cost at 2.5 hertz was comparable to the isolated leg swinging. And as a comparison, at the lower frequencies, the metabolic cost is comparable to upright standing. And we believe that the conditions, even in the 2.5 hertz, happen during everyday movements like walking, and that therefore a part of the cost of these tasks may be due to force rate. And then this is the ultimate test of the hypothesis, is we related the part of the total cost related to force rate, the force rate cost, determined it as a cost per contraction by dividing it by frequency, and related that to the peak force rate measured. And in line with the hypothesis, this is captured by a linear with significant coefficient. So the conclusion from the experiment is that the force rate cost is proportional to force rate. The force rate cost can be as large as the work cost. And we know that the force rate cost is supported by underlying mechanisms, including calcium pumping, and is not included and not predicted by Hill-type models. So what about the mysteries? We said that humans walk in a stereotypical manner with a certain step frequency, and rattlesnakes rattle in a stereotypical manner with a stereotypical rattle frequency. We start with the rattlesnakes. So rattlesnakes have this relationship between frequency and body temperature. And rattlesnakes contract their muscles synchronously with the movement. This means that on the vertical axis is also muscle contraction frequency. And this contraction frequency is similar, to our experiment, where the waveforms also were associated with contraction frequency. And so from our experiment, we may expect that there's a force rate cost with increasing contraction frequency. This is, however, not the case in rattlesnakes, as here is the ATP consumed per contraction against the contraction frequency. And when I first saw this, I was very surprised, because this seems to contradict the force rate hypothesis. But then I remembered that rattlesnakes have temperature-dependent dynamics. As we see on the upper right plot there, the force rate achieved is much higher at 35 degrees, resulting in a shorter time to peak as at 16 degrees. Other way to say this is that the time constant for the dynamics is lower at 35 degrees than at 16 degrees in rattlesnakes. And then think back to our conclusion of the quantitative model, which was that the calcium amplitude increases with force rate. But I also noted that this is dependent on the dynamics. And so if you imagine that this relationship happens with a high time constant, slow dynamics, you can increase the time constant, get faster dynamics, and the slope of the relationship is affected by this. And this means that for a given calcium amplitude, we can get a higher force rate for the faster dynamics. So a warmed up rattlesnake can get a higher force rate, but does not have to increase the calcium amplitude. And this is indeed what has been found. So here we see calcium amplitude in the warmer and the colder snakes, and they are very, very similar. And so in terms of the contraction frequency body temperature diagram on the left, it seems that the rattlesnakes prevent the force rate cost by staying on this line, staying on the body temperature frequency relation. But I would still argue that they would have a force rate cost if they would increase the contraction frequency at a given body temperature, and that they don't do this to avoid this cost. Okay, so that's a little bit complicated. With humans, it may be a little bit simpler. Humans, they adjust their step frequency to speed. And one reason why they may want to do that is also to reduce the metabolic cost, as increasing step frequency at a given speed comes at a higher metabolic cost. This looks like this on both graphs, increasing step frequency at constant speed. And well, this may be the force rate cost. And so it's the question whether this increase in metabolic cost with step frequency can indeed be ascribed to force rate. And there is some evidence, but it's still work in progress. Some of the earlier evidence is from Art Quo, who found that with a simple model, so this is what we see, the step length speed relationship. Here is the empirical relationship for humans. And if the simple model minimized a force rate cost, then the model has the same preferred or same minima at this step length for a given speed, and therefore, is consistent with the data. However, if the model minimizes a swing work cost, it's not consistent with the data. And so this is some evidence for arguing that indeed the force rate cost is what is important instead of the work cost. Some other evidence is what I already mentioned before, is the leg swinging study, where metabolic rate was related to force rate. And here it was the question whether this is due to the muscle work that was not measured. But in my experiment, I showed that the force rate cost exists independently of the work cost, and that the work cost cannot account for the total metabolic cost in cyclic force production. And therefore, these two results together seems to suggest that the swing costs may be at least partially ascribed to force rate. Okay, hopefully I've clarified these mysteries a little bit. This is my presentation. I'd like to thank my supervisor, Art Quo, for his feedback and help in this talk. And of course, I'd like to thank the funding that made this research possible.
Thank you very much, Tim, for your interesting talk. Before we get to the discussion period, I would like to kindly encourage you to turn on your web camera during the discussion period, if you feel comfortable with it. Thank you very much. If you have any questions to our speaker, Tim, please use the raise hand button that can be found under the participant icon at the bottom center of your screen. And if you click it, you will see the blue icon, raise hand button, at the bottom on the right-hand side. The floor is now open for the question. Actually, Tim, I have a very much technical question because I'm not super familiar with this field yet. So in your new algorithm to estimate the muscle work using ultrasound, video, or images, how did you choose superficial region and deep region? Because to me, it's kind of a really vague area. So I'm just curious if you could give detail or brief explanation on that.
Yeah, that's a good question. A lot of slides. Okay, so that's here. So here in this example, the top 40%, I believe, of the image is superficial, and the lower 50% is deep. And this is a little bit done on purpose because the superficial aponeurosis doesn't move nearly as much. So for a given muscle, the superficial stays roughly where it is. You can understand this is that the reason why the superficial aponeurosis has its vertical position is because of the skin and the fat underneath the skin. And of course, this will not change much. And so for the superficial aponeurosis, you can get away with specifying a narrower region without having the risk of the aponeurosis leaving that region. For the deeper, that's why I use 50%. And most of the time, this is enough. It does move, but it's very rare that it will move above the 50% line. And this is something that you can also change pretty easily in the algorithm. So I didn't mention this, but this is the more tricky part of the algorithm, the aponeurosis estimation. And this is also a new part, as previous algorithms have not really used this a lot. They did mostly Fascicle estimation. And so I would always go through what the algorithm did, like how it identified the aponeurosis, and check whether that actually makes sense.
I see. So since we are on this image, I actually have one more question on this. So you choose to use VL versus lateralis muscle. And I also know that the muscle fiber length is quite long. So would there be any reason for you to choose to use versus lateralis muscle rather than other muscles that has like a shorter fiber length, so that can be easily measured?
Well, at the knee joint, I think there are no really better alternatives. Vastus lateralis is the main muscle that has been used for ultrasound. I know, for example, rectus femoris has parallel fibers, I think, so that's even trickier. But yeah, you could go to the calf muscles. I believe one of the reasons why we did a knee extension is that the knee extensors are bigger, and therefore they can achieve, well, they have a larger effect on total metabolic rate that we measure with respirometry than with the calf muscles. And so in the lower frequency conditions, the metabolic rate is actually pretty low. And so it starts to become an issue that you have the measurement noise on the respirometry system. And so you want to maximize the amount of force, basically, that relates to the energy cost. But you don't want to have a too large force that things become anaerobic and that participants cannot do the task anymore. Because of this is a respirometry. They had to do the task for 6 minutes, which is quite long.
Okay. Thank you. Since I do not see anyone raise their hands, I'll take a chance for me to just ask more question. So I think, if I understand correctly, it seems like your model kind of overcomes the limitation that was found in heel type muscle model. So I'm just curious if you have any future studies that you have in mind for related to this.
Yeah, so one of the things that is some ultimate goal is linking this new dynamics model to a cross-bridge type dynamics model that you could call that a Huxley type model. And I've worked a little bit with this. I know that Koen, who was in my lab, in the Qual lab, he basically wrote his thesis about that Huxley model. So at some point, I hope to learn more about that and then try to incorporate that. And with this Huxley type model, you can avoid the dynamics to become super fast. I explained the heel type model. If you have isometric contraction at optimal length, you basically don't have dynamics. But in a Huxley type model, you will still have dynamics because you have the rate constants of the cross bridges that attach and detach.
I see. Thank you, Tim. I see Venus, you have a question to Tim?
Yes, actually, thanks for a great presentation.
Thank you.
Actually, I do not know what parameters you took into account in your model about calcium and how the amount of calcium changes in relation with the cross bridges. But like, have you considered taking into account the cooperativity of attachment of cross bridges? Like initially you have, or in general, the tension PCA relationship, that initially you have relatively a big amount of calcium release and the force does not change by much. And then at a given threshold, with very small calcium, the force increases like pretty quickly, because the attachment of one calcium to a given troponin would induce the attachment or the rotation, the change of conformation of the next tropomyosin and the next one, and induce a like series of cross bridge attachments. So it's initially, maybe it's true, like you need lots of calcium to induce force, but at some point there is this cooperativity that would need just a small amount of calcium to produce, to increase the force by a big amount.
Yes, this is definitely one of the things we are looking into. And I think, so as I mentioned, I want to couple this to a more realistic cross-bridge model, because now I just use the first order system, which is very simple, of course. And this will be one of the things I will consider in including it. And I think if I would use the Huxley type model as formulated by Kuhn at this point, I would need to add this cooperative cross-bridge binding, because I think it's not yet present. So I have to find a way to, to see how I can include that.
Okay, thanks.
Brent, please go ahead if you have any question.
Tim, last year at the ISB, Ross Miller and Glenn Lichwark organized this symposium where they sort of gave out data, and the idea was that people didn't know what the metabolic cost was, but they used their models along with the experimental data that was provided to them to sort of predict what the metabolic cost was. Did you go to that?
Yes.
Were there, okay, good. So were there any, my question is, did any of the models at that symposium incorporate this concept of force rate into their predictions, or are they all sort of the Hill type, Huxley based type of models?
I think none of them actually explicitly incorporated force rate, except for one person from who worked with Art previously. Maybe this was even Doki or someone else. Probably Art can help out here. So someone who works with Art, they I think has a cost, a swing frequency cost, and that is not the same as a force rate cost. But of course, we think that those two are related. And so, yeah, I think that's one of the ways that, so in general, the predictions were pretty bad of all of the models. I think that was the conclusion. And I believe the swing frequency cost is probably not included accurately in most of the models. And probably this model from Art's previous student did a little bit better there. But of course, that model was only relating to swing frequency, and that's still a step away from force rate.
Great, thanks.
Okay, John, if you have any questions to Tim.
Yeah, very exciting stuff, Tim. But my question is, there has just been a tremendous amount of work done over the decades on muscle dynamics. And yet I sort of see this issue arising out of modeling optimization of the dynamics of walking, where it's necessary to have some kind of penalty, or else everything becomes impulsive. The optimization is always defaults to impulsive. So it was a search for some penalty. Why didn't the muscle physiologists figure this out?
Ooh, maybe that they are not simulating whole body movement, so they didn't have to think about whether or not they need a penalty. Yeah, and then why didn't they use conditions that would show the force rate cost? I don't know. Well, there is a reason why you do a certain experiment, and if you're not triggered into making a certain hypothesis, you don't do the experiment, I think.
It still seems amazing to me that the cost could potentially be as much as the work.
Yeah. Well, the people that studied rattlesnake, they didn't study this because they didn't observe it. So for some animals, at least, it seems like there's no force rate cost. But again, this has to do with their dynamics. Yeah. But yeah, I cannot look in the head of muscle physiologists, so it's a difficult question.
Okay, I see Walter, you have a question as also.
Sure, yeah, thanks for a nice talk, Tim. I have a couple of questions. You know, since we talked about the rattlesnake, I think there was work by Larry Rome, at least to a certain extent. And I've never seen him present on his rattlesnake work, but I remember in 1999 in Ken or at the muscle meeting, he was talking about the swim bladder and kind of same thing. He was arguing the swim bladder in these animals when they, throughout their mating call, would be activating and deactivating about 200 times per second. And it was synchronous as well, I guess, like the rattlesnake. And at that time, he seemed to give, if I remember at least correctly, he seemed to get the idea that the reason why the swim bladder could be activated and deactivated and the force go really high and low is that somehow a minute change, very similar to what Venus was saying, except not due to cooperativity, but he found that in the swim bladder, a tiny amount of calcium change would activate the muscle really highly or very little. And therefore, the cost of calcium pumping was minute because you know, it didn't really take a lot to activate the muscle's eye to get a high force and a low force. And I wonder if other systems have evolved in a similar way. Probably not the human system, because I don't think we do anything at 200 hertz.
Yeah. Well, I think there's a lot of the adaptations that happened because, of course, these animals that contract at such a high frequency, if they wouldn't have adapted, they would pay a very high cost that we think is, well, force rate cost or calcium cost. One example is parvalbumin. Certain animals have that, which is also believed to save energy. The way that works, I think, is calcium first binds to troponin, and then it can detach from troponin but bind to parvalbumin, and so then it can be reused, so to say. So then it can go back from parvalbumin to troponin instead of being pumped out again. And yeah, I think a lot of these adaptations. I didn't know about this one specifically, and then I didn't know that the swing.
You know, Larry Rome talking about it at the symposium there. I mean, I never really read an article about it, and so, but I thought it was a clever adaptation. That what he claimed, and I have absolutely no independent proof of that, but that these tiny calcium modifications would give lots of force or very little force. And another question that I had is, I didn't quite understand, you know, like the force length velocity relationship and how that got it wrong and couldn't really, was not really predictable or predictive of the force rate cost. And at least in my own definition, I think of the force length relationship, you know, as the relationship between a muscle length and the maximum isometric steady state force. So I think about it as a steady state type of a relationship, and similar the force velocity relationship. You know, it's at a constant shortening velocity, maximum activation, active force at optimal lengths, but you are at a steady state, so the force is constant as you measure as you impose the velocity or vice versa. So I always thought about these as steady state type of relationships. Therefore, I would never ever expect that they would be able to predict transient effects. So
Yeah.
am I missing something, or would you have expected that these relationships actually should predict that? Because I would not have.
Well, that's a very, very good point. So there's nothing wrong with force velocity and force length per se. They're, of course, valid observations. The issue more is that in Hiltide models, they're not used for steady state purposes. They're just used for determining or predicting the force giving a certain active state.
Yeah, and then you're in trouble. Yeah, then you're instantaneously in trouble if you then use force length and velocity at face value and you don't, I mean, yeah. So
Yeah.
So yeah, I agree. I agree with you then. Yeah, yeah.
Yeah. So yeah, that's
yeah.
yeah.
Go ahead.
Yeah, that's really one of the shortcomings of the Hill type model. They, of course, they had to come up with some kind of model that is simple. But yeah, I think it more and more seems to show that there's just some of these aspects of the model that should be revised.
Yeah, I mean, yeah, I guess the point that I'm trying to make, I'm not a muscle modeler, but if I ever was a muscle modeler, I would not take steady state properties that are measured under very specific and well-controlled conditions and then use it to transient conditions of activation, deactivation, movement, changing forces all the time. That, I would not expect them to be accurate.
Yeah, yeah, and I think they got away with this using the activation dynamics. So I think most of the people in the past, they sort of chose that time constant that we see here, here in this case 10 milliseconds, as to match the force data. So given a certain stimulation, match the force data, because it's hard to measure all the steps in between, of course. And so that was a way to sort of compensate for this, this issue. And maybe that's also one of the reasons that they didn't think of this more as to whether it's valid to use this force length velocity type, because the result, the predicted force, seems to be can be quite okay.
Yeah, as I always say, you can get conceptually incorrect model and make very good predictions. So then the engineering outcome, the force outcome, is actually quite good, even though you might have an underlying theory that really is not quite correct. And then just another tiny thing that it's a little bit a pet peeve of mine, and that is, when you said that with the fascicle projection and seeing how that changes, you will get the length changes of the series elastic element. And I always find there is two definitions of series elasticity in muscle. And one is what I call the experimental biology or physiology one, where they do exactly what you do, where you take, you have the total muscle length and you subtract kind of the fascicle length and the angle of pronation corrected. Well, Fanningen-Schenau's 1997 definition. And then the rest is all series elastic element. And I have no problem with that. But then people like Tom Roberts and Per Agard and others, they then use the tendon force to calculate from changes in aponeurosis length, work, and elastic modulus and stiffnesses, whereas we know that the aponeurosis forces are completely different and are in fact changing and are not the same as the force in the tendon. And therefore, when you do that, you vastly overestimate what the series elastic component does. And so I just wanted to mention that because I think I have no problem calling it a series elastic element if you talk about it structurally and anatomically, but I have a distinct problem if you talk about it in terms of storage and release of elastic energy and Young's modulus and stiffnesses and that type of thing.
Yeah,
does that make sense?
Yeah, it's a very good point. It's also what we thought about, and we revised this work estimation a lot because things came up, and I read your paper about the aponeuroses, and I think currently how it's currently formulated, it doesn't make a lot of assumptions. We do call it a series elastic element, but we don't assume what it entails, whether it's tendon or aponeuroses or a combination. And it's what you said, like we assume a constant total length, and that the change in the fascicle length is due to that mysterious series elastic element. And we have the assumption that there's no external work done, so that the muscle work and the series elastic element work are equal and opposite. But again, there's no assumption on what actually is in the series elastic element. So I think currently it's pretty robust for all these concerns, against all these concerns. But yeah, it's tricky business.
Sorry, Walter, I think you're muted.
Yeah, actually, I was thrown out there for 10 seconds. I'm not sure why, but I think I got most of your answer. Because, the good work, the groundbreaking work in that area came out of your lab there in Amsterdam, with Zurbier and Hoying. who showed beautifully in the medial gastroc of the rat that when you activated the muscle and force was increasing, very often certain parts of the aponeurosis were actually shortening. Yeah. Demonstrating that you don't really have that series elastic type of behavior that you would expect. With increasing force, a spring in series, of course, would elongate; it would not shorten. And we have shown that, and Rick Lieber has shown that. So I think we just have to be a little bit. I always like people to be a bit careful. I much rather have people call it elasticity rather than series elasticity, because from an engineering point of view, to me, series elasticity has a definite mechanical meaning. That means, if I have two things in series with one another, then they carry equal forces, whereas the aponeurosis does not carry the force of the muscle and does not represent the force in the tendon. I think that's where sometimes a confusion comes in.
Yeah. Yeah.
Okay. Art, do you have any questions to Tim?
Not really a question, but I just wanted to loop back to what John Bertram asked about a while back, which was he was asking, why did muscle physiologists get that wrong? And I would say, I don't think the physiologists got it wrong. There's a subfield of people who've been studying calcium pumping pretty seriously, and that includes people like Larry Rome. Tim also had some references to people who did some isolated muscle or whole body experiments related to calcium pumping. And then Doug Syme here at U of C also has a history with calcium pumping. A lot of these groups don't call it force rate cost. That's just a name we came up with because we didn't want to give the impression that we are actually testing the calcium pumping. So we're not doing things at that level, but we're more interested in the sort of the ultimate cost that affects whole body behavior. But there are groups that have been studying this pretty carefully. But in my opinion, where things went wrong is I would say that a lot of the people who are doing musculoskeletal models and claiming that they have a model of metabolic cost, I think that's where things are wrong, because this is all based on piecing together different pieces of steady state behavior and even different animals and observations from calorimetry at different temperatures and things like that. All of that is jumbled together into these metabolic cost models. And then those aren't really subjected to real experimental tests. It's more that people put together all this data and then they say, yes, I was able to predict the metabolic cost of walking, but until recently, there haven't been many experiments. And the ISB symposium is an example of people actually trying to do an experiment where I think it was sort of exposed that these models don't do very well. So just wanted to make that point.
Thank you. Rob, do you have any questions to Tim?
Yes, yeah, I agree with Art's comments just there too. But back on series elastic elements, on your plot, first of all, Tim, I thought it was a very nice and exhaustive study, so congratulations.
Thanks.
On the series elastic plot you did of the force length curves, where have you got that? If we can get to it, the series elastic element, yes, on the one on the right-hand side, it seems to be up to 60 newtons, extremely stiff, and then it became more compliant. Firstly, was that a common finding in your experiments? And secondly, how would you explain that? And your line of best fit is not mentioning that, so I'm wondering whether you thought that was just sort of randomness. But if across your experiments, was that a common finding, and how would you explain it to start off stiff and then become more compliant?
Well, it actually starts with being very compliant because the very first value is non-zero. And so compared to rest, it increased a lot in length with very, very small force.
Yeah.
Is that what you mean? Or is it what you mean, what happens after
Well,
that, then it's stiff?
Once you get to a length change of about 0.3, then it's extremely stiff. You go up to about 60 Newton with very little length change, and then it becomes more compliant suddenly at about 60 Newton.
Yeah.
Was that a consistent finding, or is this just a one-off? Or
I would think this is just some noise that just makes it look like this. One thing I can think of is that this experiment was done by asking participants to track a very low waveform frequency target, a triangle wave. And so it might be that when you go back from, so you have a certain force and you slowly relax until zero force, that the final bit from, say, very small force to zero, they don't do that actively anymore. They just relax. And I think one consequence is that the data gets noisy there. And you really depend on the synchronization between the ultrasound and the torque. So I think this would be slightly better to just do a lot of constant torque contractions, and at each torque measure the length, because then you don't have this time-dependent thing, right?
Your confidence interval includes that sort of vertical, very stiff bit. Is this? I'm not sure. Is this a single contraction? Is this? No, it can't be. It must be an average over many contractions on one individual. What is it?
Yeah. So that's what I was trying to explain, is that this is a ramp in torque. So you start at 0 torque, and then you increase slowly until 16, and then you decrease until 16, until 0. And during this whole interval, ultrasound is measured using video ultrasound, and torque is measured, and the two are synced using a pulse. And so I'm just saying that at around 0, people find it hard to, I think, exert a very, very, very small force. And so this synchronization, any error in this synchronization may have caused some error in the, in this determining this relationship. So I think that's just an experimental error, actually. I wouldn't expect this to be real.
Right. When you're getting the signal from the ultrasound, knowing how to align the length with the force without incorrect phase delays, so you're able to verify the force for that length, particularly if it's dynamic and it's slowly moving. Do you know what the phase delays are precisely? Can you be confident that when you have a measure of length, you know the precise time so that you can match the force? Yes. And have you verified that? Because I've seen that be a real problem.
Yes, and that's why we use the slow waveform frequency. So the delay between muscle contraction and force is what this whole thing was mostly about. Is about 100 milliseconds, right? Maybe half a second at most. But the force production, so the ramp in force ramped between 0 and 16, torque ramped between 0 and 16, happened in a minute, more than a minute. So it was a very, very, very slow ramp. And so, the time was much longer than the electromechanical delay. And so, that way sync issues are not really a concern, but I'm just saying that something weird goes on when you relax from a small force to 0.
Yeah, okay. Thank you, Tim.
Okay. Walter, did you have any last question?
To the discussion, because we did in a study, we did measurements on vastus lateralis where we did exactly what Tim was suggesting, that we have people go to like 5%, 10%, 15%, 20%, and so on of maximum torque. And when you do that, then you get a very nice nonlinear relationship where it's soft at the beginning, gets harder and harder. So if I remember correctly, in the first 20 or 30% of force, you get like 70 or 80% of the fascicle shortening, and then very much less. So you do get that. And then the other point is exactly what Tim was saying when we measure in medial gastrocnemius in isometric contraction with sonomicrometry, isometric contraction, then you activate the muscle and it deforms and the fascicles get instantaneously shorter. It's a very quick response, but then it takes, when you then deactivate, force goes to zero, and it takes several seconds for the fascicle length to go back to original. I'm not really sure why that is, but it does take seconds for some reason for the length to relax when force is already zero. In the cat medial gastroc, and I assume other muscles are similar.
Okay. On that note, I would like to thank Tim again for giving his talk and sharing his interesting work with us. Next week, we will be hearing from Emily Mattia-Vish, who is a potential postdoc working with Dr. Ben O'Neig. And the title of her talk is Obstacles and Opportunities for Monitoring Overuse Injury Risk with Wearables. Thank you for coming today, and I will see you all next week.
Thanks a lot, Tim. Thanks a lot, Songwan. Thank you.
Thanks, Walter.
Very nice. Take care, everyone.
Yeah, take care.
Nice, Tim. I thought your slides were awesome too.
Thanks, Brent.
Good job. Thanks.
Thank you, Tim.
Yeah, thanks for hosting. That went well.
Hey, actually, Tim, do you see this gentleman has actually a question? I mean, I wasn't able to get him because we are running out of time. But,
Yeah, no, go ahead.
yeah, but Brent, if you are around, if you...
Yes, I'm around. Sorry, I don't put my video on, but it's...
Yeah,
midnight.
no, please. This is
I'm in my
sorry.
pajamas. I just wanted to ask you actually regarding to the last question about this weird series elastic element change where it gets stiffer than compliant. And you can directly see it occurs because of the fascicle length change. If you just tilt your head to the right, of course, those two are basically mirrored. And I think it's actually a problem with your tracking. And that's a problem with the method you're using for the tracking. And that is that you're not tracking the same fascicles in your contractions. So the fascicles from one end, say the more distal end, are coming to the part that you're imaging with ultrasound.
Yeah.
And so the orientation of that part of the image can actually be completely different to the initial part of the image when you weren't contracting. And that's why UltraTrack was actually made. So you could track the same fascicle. As you found, it's not very good when you have fast movements. It's really good if you do things like postural sway or passive rotations. But unfortunately for your kind of movements, I actually think if you check in MATLAB, there's a vision toolbox you can use. And Drazan is a guy from Penn State in combination with Josh Baxter. They have a free algorithm you can play around with. And I've been using it. And actually, it's a feature-based algorithm. It tracks fascicle length change during fast contractions quite well. So I really recommend you have a go at that.
Yeah, sure. Can you maybe send it in the chat or something? Or
yeah, sure. I will. I'll actually just send me your email, and I'll send you an email tomorrow.
Oh yeah, it's on the first slide.
Ah, okay.
If you go to the first slide.
It's actually really by chance so that I even knew this talk was on. So I stumbled across one of your preprints, and I'm tracking some Fascicle
Okay.
data at the moment, and I played around with your algorithm. But the best one, as I said, is this one from Drazan. So I'll email you.
Yeah, I'm definitely interested in comparing against other algorithms.
Yeah. And there's actually another one I can send you too. It's really similar to yours. So it does a similar transform, Hoff transform. I'll send you that as well.
Which one is that? Which author?
It's another guy from Penn State. Let me give me one sec. It's by Ben Infantolino, Pennsylvania State University, 2012. So yeah, for whatever reason, I have that one too.
Okay. Well, I think it's very interesting to compare, especially like I think the difference is called feature extraction versus feature detection.
Yeah.
Well, basically
exactly.
just one is just, so my kind of algorithm is just directly mapping Fascicle length to an image, whereas with the optic flow type, what I said, it's history dependent. And well, maybe that's fine. Maybe it has these advantages, what you mentioned, that it's easier to track the same Fascicle. Although, of course, that's also with within certain limits, because if your Fascicle of interest leaves the screen altogether, then
yeah, exactly. Then you're just guessing again.
Yeah.
Yeah, I mean, ideally, you probably almost do the automation like you've done, and then you implement some feature-based tracking with optic flow to try and track the same Fascicle. And then you could compare it theoretically to something that's automated.
Yeah. Yeah, and so sure, and like it would just be great if there's more sharing like this, and then we may
for
say,
sure,
we
yeah.
have this very fast contraction, now we use this algorithm, or we have this very slow thing, oh, then better use this algorithm.
Yeah.
Choices instead of choosing always the same.
Yeah, what I'll do is I'll send you an email with a bunch of details with the algorithms and some videos that I've shared on Google Drive with you.
Yeah.
Because I agree with you. It's better to have open communication and really try and find a good way to track ultrasound images.
Yeah, for sure. Well, thanks a lot.
Yeah, and thanks for the talk.
Yeah.
It's really interesting. Okay.
Thanks.
Enjoy your afternoon. I'm off to bed.
Yeah, sounds good.
Have a good night. Okay, thank you very much, Tim, also for just staying a little bit longer.
Yeah.
Okay,
No
if you
worries.
okay, I'm gonna just quit the Zoom and I'll leave you in peace.
Yeah. Yeah, I think you mentioned this, but you recorded this?
Oh yeah, it has been recorded, and then now the video will go to Hoa, if you know him. He's working on the editing video, like cropping the edges and then properly edits, and then like more editing thing. But yeah, so once he's done with the editing, then I think yeah, and then I'll let you know once it is done, and then it is uploaded into the website. But it should be on the our HPL seminar website, and if you go to watch previous videos, then that will be where you're gonna be show up.
Okay, sounds good.
But yeah, just give me some like a few weeks to be uploaded it. But yeah, I'll definitely let you know.
Okay, sounds good. Thank
Yeah,
you.
thank you.